Okay, so I'm just going to go through our gene expression practical and discuss the answers. Now I know that you can't see my results, but when we need to look at them, we shall look at them. And we will not, but I'll show you. I'll read out what kind of values I have. So for 9i, the question asks us, or it's saying, the CT values for each of the 3 reactions you have prepared. Oh, is it missing something? Okay, whatever. Does the template control sample give the expected results? So, first of all, you kind of need to know what a no template control is. So in our experiment, I mean, if you read the sheet, and if you don't have it, here you go. You can pause it where you need to pause it. I'll quickly just go through it. So that's what it looks like. Our control was this one here. And so what can you see is different about our C, our control, to, let's say, A and B? Well, hopefully you can see that actually here, this is the difference. Our no template control has no template, so it's containing no cDNA. Now, if we go back to the question, it says, does the template control sample give the expected result? And so for me, I didn't get a CT value for either of my controls, and that's what you should expect, because if you know what a CT value is, then that should make sense. And if you don't, well, a CT value is essentially the number of cycles it takes for the threshold fluorescence to be reached. So it's really the time it takes for the threshold value to be reached. And hopefully that makes sense that you shouldn't get any value for this, because we don't have any DNA in our sample. And the thing that we're using to measure the fluorescence, or to mark, really, to use as an indicator of the progression of our amplification process, is cyber green. And cyber green is essentially a probe that binds to all dsDNA. So that means it's going to bind to our cDNA, which is useful, but because it's extremely non-specific, it means that it's also going to be binding to other things that contain dsDNA. So things like genomic contaminants. So that could literally be you, perhaps, I don't know, hair, or spitting into it, or contaminating the Eppendorf tubes, because it is quite a sensitive experiment, right? Because we're using PCR. So it's going to bind to any genomic contaminants, it's going to bind to any cDNA if we have any, and it's also going to bind to any, if the forward and reverse primers are complementary to each other, this is known as a primer-dimer. Now we would expect, obviously, in the lab that we are purchasing well-developed primers, so we sort of knew that we weren't going to have primer-dimer formation. But that's something to look out for. So the reason that we don't get a CT value, all in all, is because there is nothing. We shouldn't get a CT value because there is nothing for cyber green to bind to, so it's never going to reach threshold because there's nothing for the cyber green to bind to. So yes, we got the expected results, we got the expected CT of no value because there was no template present. So no cyber green will bind because there is no DNA present in our sample, so we'll never reach the threshold value. What's interesting though is that for my practical, I actually got a TM, a melting temperature. So this is the melt point, is where 50% of the DNA becomes single-stranded, and this is kind of strange because if there's no DNA present, then we shouldn't be getting a TM value, but we did, so that's just sort of a spurious result. It's something, it's a questionable result. We're not quite sure why we got that. The next question says, does the melting curve for the 3 reactions you prepared give the expected results? What is the purpose of the melting curve? So like I say, you can't see my values, but they do give the expected results. They do give the expected results. So I'll see if I can find a way to actually link my results, because that might be a bit useful. But yeah. So what is the purpose of the melting curve? Well, the melting curve shows us the CT values, and the different curves that you see on a melt curve actually shows us the differences in the initial cDNA in the sample. So you know that we used A and B, where A was our untreated cells and our B was our treated cells. So basically, the longer, the higher the CT value, it means that there was more cycles needed for that sample to actually reach threshold. And that would indicate that actually there is less cDNA in that initial template. Hence why there is a longer cycle time, or why there is a higher value for the cycle time. And so when you plot your graph, so they haven't supplied a graph, but if you go through your notes, you'll see that the graph sort of has a shape like this. I'm going to try and outline it with my mouse. But we start with a very flat line, and the reason that we start flat is because obviously we have done very few cycles of amplification, so there's going to be hardly any cDNA present. And then when we reach the threshold, that's where we actually start to enter our exponential phase of growth, and that's where we get a huge increase in fluorescence, and that's because obviously the amplification enters the logarithmic stage of amplification. And again, when it plateaus you'll see it later on the right hand side of the graph, it's going to plateau because perhaps some of the reagents have been used up. Alternatively, the Taq polymerase might just stop working. So there's always that. Then we have, what do the CT values for PCR reactions A are untreated and B treated indicate about the cyclin D1 expression in the two samples? Well, so my CT values for A were roughly about 22, let's just say 22, and the values I got for my B sample, my treated sample, were 19.01 and 19.75, so roughly 19.4 as a mean. And so you would expect that the treated cells, by the way, are cells that are being, when we call them treated, we mean that we're using epidermal growth factor. That's what it means by treated. So we use, my B values, like I said, were around about 19, and my A values were about 22. And that to me shows me that there is actually a greater level of expression in our treated cells because the CT value is lower. So it took less cycles, it took a shorter cycle time to reach the fluorescence than it did in the A. So what does this indicate about cyclin D1 expression, or cyclin D1 cDNA in the two samples? Well, there is more cyclin D1 in our treated cells. And what can you conclude regarding the regulation of cyclin D expression by epidermal growth factor from your data? So again, epidermal growth factor is what we're using to treat our cells, so that's what we used for our B cells. And so from our results, because like I mentioned in this answer for part 3, our B values were a lot lower, our B CT values were a lot, were significantly lower than our A values. And again, that means that we reached our threshold value, before we did for A, and that means that there is a greater expression of cyclin D1 in our treated cells, which would indicate actually epidermal growth factor is increasing the expression of cyclin D1 in the treated cells. And so our final question is, what controls should ideally be incorporated to validate the qPCR results? Well, there are a few different things that we can do here. We've already mentioned that we used a no template control, and that's where we use no cDNA. The next thing that I'm going to say is sort of redundant because we used CyberGreen, but I'll mention it in case there is a question where we don't use CyberGreen. We could use a no Taq polymerase. We could use a no Taq if we used the Taqman probe, but we didn't use the Taqman probe, so again, that's redundant. If you don't understand that, then I can make a video on the 5 to 3' exonuclease activity of our Taq polymerase and how that functions with the Taqman probe if you'd like. But anyways, that's not important for this video. We could also do a no reverse transcriptase. So this would be where we use our mRNA and we essentially don't make any cDNA because we can't make cDNA from DNA polymerase because it can't use a RNA template to make copies of DNA. So essentially for this then, if we use no reverse transcriptase, then we don't get any cDNA for PCR, so there will be no amplification. And a final thing would be we could use a reference gene. So the reference gene is basically something that the expression remains constant in both of the samples, and that's because it's expressed in both samples and it's not going to be affected by the treatment. So, for example, epidermal growth factor won't change the expression of something like GAPDH, glyceraldehyde dehydrogenase, or something like that. Actually, I don't think that's glyceraldehyde dehydrogenase. I'm not sure what it is, but GAPDH, because the expression will be constant in the untreated and the treated cells, so it'll allow for correction if there are differences in the CT values. But these CT values for the reference gene should be exactly the same in both our untreated and treated. So hopefully that was useful, and yes, that's all I have to say.